Pharmaceutical Processing, in conjunction with Interfex 2018, presents Interfex Live.
All right. So as promised, we're going to talk about biosimilars. So we have with us today, we have Mike, and we have Hallil, and Rick. Okay? We're going to get right to it. Okay? So, the reason for the name, Biosimilars 4.0. If you've come before, every year we do this, and every year it's not only is the most interesting, it turns out to be the most well attended, and more importantly, it's the one that has the most change. So, not to disappoint anybody, here we are again. Okay, so what we're going to talk about is where we're going to start in the usual spot. So, Rick, I'm going to look to you and say, what do you believe that we have made more progress in the area of biosimilars this year? Bring us up to date.
Yeah, I think. Can you hear this? Okay. I would say that there's been a huge advances in biosimilars and approvals. Europe, especially, probably 16 approvals, 17 approvals in 2017. More on the way for 2018. FDA not quite so active in approvals. We're getting there. So I think there's been a lot of movement towards the good.
You could chime in if anybody other than, let me ask you this. So what have been the challenges?
Well,
go ahead.
Okay, in terms of challenges, what we have to do is the FDA has to get their pathway nailed down. There are a whole bunch of guidances coming out there. One of the challenges, we see products coming out there. Being approved, however, is not the end of the game. The end of the game is getting launched, and not even getting launched, but getting used. So we're making progress with the FDA. There are 60 programs in the biosimilar development pathway. The biosimilar user fee has been accepted. People should have minimized that because there, for the first time, biosimilars now stand on their own financially with our own metrics. And we're measurable. The FDA is measurable. We're measurable. The biggest challenges we're having right now. So we made progress, but we haven't yet. We only have 3 products on the market right now, and 2 of them are struggling considerably on the market. So the big challenges that we have to overcome are related to getting the products accepted by the public at large, getting them accepted on the formularies, and finding a pathway through the patent barriers.
Right. Well, while we're on that subject, so we see a lot of terminology out there, and sorry about that. I've got to get myself resituated. So, what is FDA talking about when they talk about totality of evidence and the approach for developing an approval? Let's step back and talk about that.
Okay. Totality of evidence is a term being used for biosimilars. It actually means something different than what it used to be for other drugs. There's no single piece of data that's considered to be definitive. Phase 3 programs as a definitive efficacy and safety, you don't quite have it the other way. For biosimilars, you're starting with the ground level, which are the analytics. If you don't have a product that really highly matches, game over to begin with. Go back, start all over, make sure you have a product that matches well. You have to do the analytics, you have to do the functional, that's the bioassays. Then once that is shown to be comparable studies versus a reference product, multiple lots that you match, then you go to the next step, which would be the animal studies. There, it's certainly abbreviated compared to what you've done for developments. Then the analytical program is actually different again. Now the most critical studies are actually the pharmacokinetic studies, and if you have pharmacodynamic studies, those as well. You do those in healthy patients because they can actually show bigger differences. Then once you have that, you have to show immunogenicity. The FDA and the EMA may give you a pass at doing a comparative safety and efficacy studies. They've done that actually for 2 products so far. Most of the others have had to do it. What happens is that it's possible to fail 1 or 2 assays or 3 assays or come close in your pharmacokinetics. I know that one of our products actually failed the upper bound of, let's say, 1.255. But they say, based on taking everything together, the totality of everything together, we feel that you have met the standard of high similarity that is required. So there's no single assay, but saying everything being put together. That makes it difficult for companies developing the products because you may have a different opinion than the health authority. But you really have to come really close.
But just to, as a comparison, if you're trying to come up with a generic and you don't put it between the goalposts on a crossover by a BE study, you're out. I mean, so it sounds to me like maybe it's more scientifically based, a little more stretch.
It's not the stretch, because you have the analytics are where it starts with. But it's possible to miss on one analytics. It's also possible that if one of the quality attributes that's not critical doesn't match, that's another question as well. But what we're saying is it's not bioequivalence. Bioequivalence is just probably one of the last pieces of data. Now, if you don't meet bioequivalence, it's going to be very hard to meet the totality of evidence, realistically. If you come extremely close, like 1.255, perhaps. If you're 1.45, you don't stand a chance. But each data has to be considered on its own merits. So a failure for bioequivalence may not be the end, but it really depends on how close and what the parameters are.
So Mike, let me ask you this. So what is different about the manufacturing? We're talking about biosimilar protein compared to a new one. I mean, so what are we talking about as far as what effect does the size and complexity of a protein have on the overall development program? Okay.
It makes a number of differences. When you're making an innovator or a new drug, what you're starting from scratch. You're not comparing against something else. The biggest hurdle, and it's a significant hurdle for a biosimilar, is getting that comparability to the reference product, to the product that's on the market, innovator. And as Hallil said, you need to acquire a lot of lots. Typically, I would say at least 20, some people up to 100 lots of the reference product, because within any biologic, which is different from the chemical drugs, you have a group of closely spaced relatives. There are slight differences in charge or hydrophobicity. They're just part of the way proteins are made through recombinant protein technology. And so this has to do with degradation of certain sensitive amino acids, differential post-translational modifications like glycosylation. This is a very prominent post-translational modification. And if that's a little different, the molecules are going to show a range, sort of, I think, what you said, Russ, bookends or so, for each of your analytics. And analytics are dominant in biosimilars. They're very important in making a new protein, but in biosimilars, it's almost working like somebody looking over your shoulder. All the time you have to compare against the reference or innovative product. Because if you're outside of that range of critical quality attributes, those are the elements of the molecule that defines, for example, its mechanism of actions, some key chemical properties, some effector function on some antibody-based drugs, and so forth. If you're not addressing that with close comparability, then you won't make it as a biosimilar. You may make it as a new drug, but you won't make it with much more work. So what's constantly done in the biosimilars is this extra burden of comparability. Now, the payoff for that is your clinical trial burden is much reduced. Your safety and efficacy are pretty much assumed because you're making the same protein, similar protein. If your family of protein is close to the other family and all these critical attributes, then you can push through and you can jump forward much faster than you can and with lower cost than you can for a new product.
Okay. Yeah, Hallil, go ahead.
There's an interesting aside I think our audience would be interested in. We have already found out things about the reference product by doing these comparisons that were not known before. I'll give an example because it's been published. With etanercept, we did the analysis and we found out that there was a loss in potency in the assay, and we tracked that down to a misfolded protein. This actually was something that was not known with the reference product. But now it was sort of interesting that the potency had been declining over time. It was still within the goalposts, but when we tried to do the statistics, there's always a problem. Why is it declining? And we don't have the same problem. What we found, it was the misfolded proteins. We were able to show how to control for it. I'm not saying there's any problem with the reference product, because obviously it's been used successfully. But it's the first published situation I'm aware of where a biosimilar manufacturer has shown changes that had not previously been known with the reference product. But there are other examples as well that haven't yet been published So this is part of the totality of the comparisons. You start learning things about the reference product that had not been known before.
I got you. So Rick, why don't you talk about how you address some of these challenges?
Yeah, well, one of the things that we look for is to make sure QBD is being done between the 20 lots, so there's good similarity and there's able to show lot-to-lot similarity, obviously not identical, and then also building a process that can be put together that is a smaller footprint, continuous, and more able to give control. I mean, that's not everywhere at the moment, but definitely that's a trend towards some of the ones in Europe and Asia. We see that more and more starting to take shape, and definitely there's a link into PAT, to QBD, and then the control of the whole process of making biosimilars. With that, it hopefully addresses some of these concerns of having a consistent product.
Got you. So Mike, you're coming up, but I just want, it's not unusual, you know, where I know in the small molecule area, it's like there's physical chemical manifestations that were not identified before, which in the end result in the next guy finding this out, much to his chagrin, because it usually results in problems. Yeah, so I think that just shows the maturity of the science, and I think the quality of the science that's being done. Let me ask you this, Mike. So, did you want to comment on that?
No, I think you're absolutely right about the advances, especially in analytical technology and certain things that like mass spectrometry can do today, which is resolution to 1 AMU, which is a 1 part in, say, 150,000 for a monoclonal antibody. Yeah. It's just astonishing.
So let me ask you this question. So I want to get started. I want to develop a biosimilar. So what are we going to do? Am I going to IND? Why don't we just step through it real quick? I mean, what do you think?
Well, I mean, first you're going to choose a product that you're going to make a biosimilar. That's almost a separate discussion about how to look for a place where you're going to look at competitor analysis and so forth and market analysis. But stepping aside from that, assuming you've made the decision you're going to make a biosimilar, then you need to fully understand what you're going up against. What is the reference product? And not just, the basics, but you got to characterize it in great depth and through multiple lots, because each lot will show a certain variability from the previous lot. And you need to find out how much range each of those critical quality attributes has. And then you have to understand the mechanism of action of the reference product, to help understand what is critical and what's not. Now, as Hallil alluded to, there'll be some attributes that are not critical, but they're different. And if they're different, you're going to have to say, well, why is this not an issue? Because my product has, a little different glycosylation, but the glycosylation profile is not so important for this nature of this drug. But it's not just the structure, but it's also its mechanism in the patient. And this is the PK and PD studies that Hallil referred to earlier. Where what you think is not so important to say for binding the target or for effector function or for enzymatic activity, it may have an effect on the elimination profile of the drug. So your dosing could be different. So all that has to be carefully considered.
I got you. So Hallil, why don't we talk about that extrapolation aspect?
And the extrapolation is actually an interesting part. There's been a lot of education in that area. Most healthcare practitioners, patients, think of extrapolation as one indication to another. And we saw this with the first AdComs, but that's actually not true. Its extrapolation is from molecule to molecule. And in fact, the FDA just put that out there actually last week. They had a Reddit survey with Leah Crystal, and that's what you said. You start out with showing the molecules are highly similar from an analytical and functional perspective. You go to the next step in the totality of evidence, which is non-clinical. You go to the next step, which is pharmacokinetics, which is what happens to the drug in the body. If perhaps pharmacodynamics, what happens to the body and the drug. You do those in a sensitive population so that if there's a difference, you'll be able to see it. That's why we tend to do pharmacokinetics in healthy individuals, not in patients. Because healthy individuals are not compromised by the disease status. They have fully functional immune systems. So if there's a difference in immunity, you would see it. If the structure is the same, structurally, functionally, pharmacokinetics are the same, pharmacodynamics is the same. You've seen no differences in safety and efficacy. Effectively, you're saying then, and you've looked in the most sensitive or a very sensitive patient population, it's safe to extrapolate and say the patients in indications perhaps less sensitive will also respond in the same way. Essentially, the same molecule will have the same response. So you're extrapolating the molecule itself. You're not saying the indication, clinical indications are similar one to the other. If you think about it, psoriasis, rheumatoid arthritis, inflammatory bowel disease, very different diseases, but their underlying mechanism is the same. They both are caused by inflammatory manifestations. So if you do it for the anti-TNFs, they measured the sensitive population was psoriasis. Why psoriasis? Two major factors. One, you can actually measure on the surface of a person how much is impacted. And secondly, and also important, people don't take confounding medications such as methotrexate. So because it works well in psoriasis, we will say it will also work well in rheumatoid arthritis, even though rheumatoid arthritis is a larger population, but it's harder to measure and it has confounding medications. So extrapolation is my molecule to molecule. And it's actually pretty well established in, we don't do studies, whenever we make manufacturing changes, we don't have to go and redo the studies in every patient population. If you have to do a clinical study, you do it once, and you assume it works in the others. That's what extrapolation is about, a molecule to molecule knowledge, so that, and you know the mechanism of action is the same for different clinical manifestations, therefore you can assume it will work in the other clinical indications that have the same or similar mechanisms.
So Mike, let me ask you that again, all right, which is where we get to, and that is, how close has this thing got to be? You know, like I keep saying,
Okay.
I don't mean to, coming from the small molecule, I got the goalpost, guy.
It's actually interesting because you have to remember different health authorities may have different opinions.
Right?
Yeah.
Okay. Let's talk about the one that matters. Go ahead.
Okay, well, the FDA, of course, is a little slower behind things. Alel and I were speaking earlier about, it's about 10 years behind Europe where they are in biosimilars. But that being said, they're playing catch up, they're doing some of the right things, and we're pleased to see the progress. How close do you have to be? Well, you have to, as I started to say, you really understand your molecule, your protein, measure mechanism of action, what key degradants are, what impurity profiles are, and then you look at what would make a difference to the mechanism of action of the protein. And some elements are sort of obvious. You don't really want more aggregation. Aggregation is generally always a bad thing. So if you have less aggregation than your reference product, that's generally favorable because that's a trigger for immunogenicity. And if you're at least as good as the reference product on aggregation, then that's reasonable. But there are other critical quality attributes that relate to what that molecule does. Binding to the target I mentioned earlier for proteins is over. But one of the differences when you're making a biosimilar compared to a new drug is that when you're making a new drug, you're going for the highest titer cell line, the best productivity, most efficient process when you're picking your initial clones. When you're doing that after your initial transfection, you're picking clones for a biosimilar, you may have to get rid of some clones that are great producers, but they're not producing one that's similar in those attributes. And if you're not sure, well, you just try to pick the clone that'll produce the very closest match. And because of the powers of high throughput screening, you can do that. It's because of some of the advances we talked about earlier in analytics. You can look at thousands of those clones and find the ones that really match up on the cellular level to your. Well, you still have a long way to go because you have to go all through the development, but you have to start with a cell line that's very close to producing similar to what the innovator product, reference product is. And as a general rule of thumb, you would match the type of cell line. In other words, if it was a CHO, Chinese hamster ovary, you'd match that rather than switch to a human cell or a murine cell or something. Just because if you do that kind of switch, the hurdle's that much harder for you.
Wow. Wait, I'm going to just loop back to that. Rick, you had a comment
Rick,
there?
I just wanted to just add to that excellent comment is that once you've done all that optimization and picked the right cell line and got as close as you can, the process also needs to be made sure that it's dialed in and it says modified and made to the point where it can optimize your production, give you the most antibody or recombinant protein possible. And you do various screening techniques by taking different parts of the chrome process, the purification process. You can run a DOE. You can cut down on a lot of the work as well before getting to that final process, which allows you to shrink the footprint a little bit, tailor it, so you can take some of those costs out, which are very key to biosimilars when they go up against the major drug.
Yeah. I might be able to answer your question directly. How close is close? I've worked on 5 products already with the FDA. Step 1 is to agree on the attributes you're looking at. What are the critical quality attributes? And then there are 3 categories: most critical, middle category, and less critical. And those that are most, and you sit down with the FDA, you propose what you think it is. The FDA then tells you whether you're right or wrong, they disagree. Agree or disagree, that's probably what I meant. And then you set the ranges. The FDA decides whether they agree with them or not. Then it gets a question of, they want 10 lots for analytical statistical purposes. There's a draft guidance on analytical statistics. And depending on the category, you have to apply one type of statistics, observational or just within the range. So it's how close is close. Step 1 is to identify the critical quality attributes, reach agreement with the agency before you do the assays. Then you have to apply the statistics that are specified in the guidance, which incidentally a lot of people in the industry have challenges with. That's completely a different situation. But either way, you identify, you agree on the statistics, you agree on the number of lots, and then you have to try and meet it.
So this agreeing on terminologies, let me ask you the next question, which that's the biosimilar versus intended copy. I mean,
that's really something else, and that's actually very interesting. There are, around the world, there are different standards. There are companies and there are countries where they say there are products that are biosimilars, but they are not head-to-head comparisons against the reference product. So they would not be considered as biosimilars as in Europe or the US. I'm not saying that they're bad products. I'm saying that they did not have the comparison, head-to-head comparison, as expected by the World Health Organization, by the Europeans, by the Americans. And if anyone wants to know, you'll take a look. Last week, Colombia proposed a 3rd pathway, which we would consider a intended copy, where they're saying if the reference product is well known, all they're asking for is a pharmacokinetic study. Well, it's intended to copy the reference products. And you know, in countries like that, maybe where access is really an issue, and if the product is safe and it works, okay. But we shouldn't be calling those as biosimilars. So around the world, you have places where they're not doing head-to-head comparisons. So I wouldn't consider those to be biosimilars. Again, I don't want to disparage them, because they might be fine. But a common term is they are intended to copy the reference biologic.
Okay, well, let me ask you this, Mike. I mean, this is something you had mentioned before. Forgive me. I mean, to me, I just try to get my head around the terminology, biobetter. Let's try that one. What's
okay?
All right, let's talk about biobetters.
Sure.
I mean, now what's that,
Now, biobetter is a very closely related molecule to your original or innovator product, but it's got a better quality. For example, it might have polyethylene glycol or PEG attached to it to give it a longer half-life. It might have a better effector function, so in the patient, the immune system is more potently activated by that. It's got, in the case of that type of product, an antibody. It's got an enhanced glycosylation pattern that allows stronger effector function, and so that is known as ADCC, and there's ways to make that greater or stronger. It's a 2nd generation product, but it's no longer a biosimilar. So you have some advantages over a brand new product because you know the indication, you know basic properties of the protein. It's been in all these different patients in a slightly different form. But you still have the full hurdle of a new product. So you're filing a complete new biologic license application. It's not a biosimilar. So you have to go through the the full clinical trials. You don't get any pass on all that, but
got you.
you know a lot more about that type of protein. And you know, I have worked on one that had modification of 3 amino acids out of the large protein to knock out a troublesome side effect, a bleeding side effect that was caused by this particular drug. And by changing those 3 amino acids, you had a new protein. For a biosimilar, you can't change a single amino acid in the chain. Everyone's got to be the same. But it was a biobetter because it had a similar mechanism of action, but the key one that was a troublesome mechanism was knocked out. And so that is still in late phase clinicals now. I'm no longer working on that, but something in my past I'd worked on. And there's a number of other ones that are even marketed, you know, with, as I mentioned, the PEG products.
I'll get to you. Just one.
Okay.
I just had one question here. So, you know, my former life, Novartis / but anyway, my question is this:
I've heard of the company.
how can I say this? Oh yeah, we used to fixed combinations are a thing, aren't they?
Yeah.
Right. So anyway, we worked on it was a blockbuster, and we came up with a combination where we found that the tag-along drug, we actually we made it twice as bioequivalent. In other words, the same dose, we got twice the area under the curve. So to me, wow, right? Scientifically, that's a great thing, right? But it's a bad thing because you got to reset the whole program.
Yeah, it's not.
right?
It's no longer a biosimilar, but it's an improved product. Now the challenge for you is that going to be better enough to make people switch from the product they know to the new biobetter version.
So half dose is not probably good
I mean,
enough.
that may not be enough of a draw. You know, now if it has less frequent dosing instead of once a week, every once a month or something, yeah, that, especially because the biologics are generally given by injection. They won't survive a GI tract, so they're not tablets and pills. So you have to, this frequency of injection matters more than whether you take 1 pill a day or 2 pills a day.
I got it. Okay, Rick, I'm sorry, I cut you off.
I was just going to come to is like biosimilars, biobetters, biologicals. You're still going to have to do a lot of PAT work, a lot of screening, LC mass spec. Someone brought up that's moved on a long way from it was years ago. You are now able to build up a track of data to see how much is the range, because anything biological, batch to batch or run to run, you can get some variability, similarities. I'd rather to use the word similarities, even with biobetters, biologicals, biosimilars. So with that process control, you're then able to put a package together which says where your bounds are.
That sounds like it's a pretty big shopping list. So
Yeah, there actually are, people should realize there actually are licensed biobetters already. Pegylated filgrastim, which is Neulasta versus Neupogen. Epoetin, where you've modified a couple of amino acids and you've changed glycosylation. Epoetin, darbepoetin, or pegylated epoetin. Insulins, you have the short-acting, the long-acting. All of these would fall under biobetters. But think about it, for every one of those products I named, the reference product, it was not a comparison against what existed previously. It was a complete standalone development program. Much larger, actually, animal testing, because you have to redo all the animal testing, have to redo the safety and efficacy testing. So, but that was done, and trust me, for insulins, it's a real advantage to have long-acting and short-acting, and those are effectively biobetters of what preceded it.
So it sounds to me like you got to do all the analytical quality characterization.
It's a new product. That's
Totally
what
new
it comes down to.
product.
It's a new product.
I
Rick.
have a question for the panel, really. Is because of a lot of approvals in Europe and others now for some Asian companies, and they are coming in the US as well. I was meeting with the FDA personnel yesterday. They are really trying to promote approvals of the biosimilars. How do you see it going then over the next few years? Is it still going to be slow, or is it going to speed up? I mean, they seemed like they would genuinely want it to go faster.
All right, I'm heavily involved with the trades and with the FDA. It's almost like a game of whack-a-mole.
They were very keen to move forward.
The FDA is moving forward, I think, and to their credit, but recognize the FDA was a really long time in developing guidances. BPCIA of 2009 was signed in 2010. The first product didn't get approved until 2015, and we still don't have all the guidances. So, so they want to move forward. they are moving forward a lot with education. That's good.
Yep.
The patent issues are more of an issue now than data exclusivity, so to put that aside. What you have is, at one time, people were saying that products have to have different names, or they're not interchangeable, they're not safe, you can't switch. I mean, I was heavily into the switching story. You shouldn't be switching. Well, now we actually have evidence that you can switch. Well, can you switch from biosimilar to biosimilar? So it's always, when we've answered one question, another one is being raised. That's not an accident. I think what's happening in Europe, think about it, the anti-TNFs were accepted much faster than the biosimilar filgrastics.
Right. Yep.
And the most recent class, which are the rituximabs, the oncology biosimilars, are much faster even than that. In some countries, within less than a year, they're now the majority product. So I think if you look at the European experience, when people and doctors become more comfortable, they get more willing to accept it. If you'll take a look at the product we have, the first one now, filgrastim, we now have 34% market share, which is actually pretty good, because the market leader is at 45. Now, there are relatively few physicians out there who would not say that they're comfortable using the product. That wasn't the case 2-3 years ago.
Yep.
So I think you have the acceptance hurdle, and that's a matter of education.
What about the movement towards like therapeutics and fast-tracking treatments through? So like oncology, MABs, and things like that.
You know, biosimilars do not, by definition, will not get fast-tracked.
So that,
All these fast-tracked mechanisms to move faster, they just simply don't apply to biosimilars. In fact, biosimilars just took a 2-month hit on the review cycle. It used to be under PSUFA 1, it was a 10-month review cycle, but we also had a 40% rejection rate at the FDA, some of which were 3-month extensions. So for the user fee extension, the FDA asked for an industry agreed for a 12-month clock now instead of a 10-month clock, and we have because the idea being we get more interaction with the agency, but the end result is there are no accelerated pathways that we get that new drugs have.
So Rick, let me just back up. So Mike, all right, here's Mike Key. Hill brought up the patent thing. So I'm sitting in the bushes. I'm looking at the innovator. So before he falls off his cliff, how much of a jump do I need to get on this guy?
Well, it's hard to put an exact number, but a minimum, I would say 5 years, but 5 to 7 years. One of the important things about the patents is that there's a primary patent for the substance, the chemical substance, but there's a whole lot of process patents that may or may not be enforceable, but the creator of the reference product has backed up the process, and they usually have a longer expiry date because they were filed later. So you have to ensure that you're not infringing in that area as well. So the reason for the advanced backup is to really get all the comparability and analytics under your belt, and getting through some the preliminary studies you need to do.
They got it. That sounds to me, yeah. So you really got to be looking way out. I mean, we were talking about this in intelligence. The young woman that was here before. I mean, to me, is you can be misguided pretty quick.
Yeah.
I mean, the bottom line is you got to know where you want to go. So, talking about where we want to go, Rick, I just wanted to ask you a question. I mean, listen, with all this work, so where are we at with this? We were talking before about this continuous process.
Yep.
These kind of dropped a couple of these out there. So, I mean, here's where I'm at. I mean, look at all the stuff that has to happen on a batch process. So, where are we at on continuous then?
Continuous, you still got to do the same PAT, and it actually enables you to be more informed about the process from start to finish. You can pick the point wheres to do PAT, what attributes you're looking for, which criticals to quality you want to measure. The good thing about continuous is you don't have to make a whole batch before finding out whether it's good or bad. You can interrupt at times and start again, which obviously you then don't end up with a whole tank of stuff you can't use, which from a cost point of view is very critical. The other thing is with continuous, you can make it as big or as small as you want it. It's very scalable, so as personalized medicines and personalized therapeutics come in, you are going to be able to do a small footprint or a large footprint, just depending on how large a market it is or how much is needed for the actual population at risk. So you can dial it in, whereas if you go for complete batch, That's going to be a very large process, may take up a lot of capital, may take up a large footprint. But there's always going to be a place for both. Did I answer your question? Or?
Yeah, I think you did. But to me, it's always a back and forth with, continuous is a trade-off versus batch. I mean, what's your sense on that? I mean, considering all the background we got to do.
The challenge is that I understand and I agree with what you're saying, the value of continuous. The problem is the health authorities, and even advanced some health authorities will accept the continuous process, but at the end of the day, many of them still want to see discrete batches, and that makes it a bit of a challenge. I'm not sure the regulatory aspects have caught up with the process science, because they want to see X number of batches to be analyzed and compared to the reference product.
I would just say from comments from yesterday, we had a meeting with the FDA, is they're looking for ways to speed up continuous because of the need to cut costs for therapeutics, especially for the sort of smaller volume personalized ones, is if we can come up with a way of showing that QBD is critical to quality and showing that the batch is okay, then they're very keen to drive this through quicker, not slower.
Yeah, I would support that because I was part of a session about a year ago sponsored by Bio New Jersey on continuous processing for biomolecules, and we had an FDA speaker, Sarah Arden from the Bureau of Biologics, and they're very supportive of the concept of continuous processing as all the way to the top from Janet Woodcock on down, and she's spoken at continuous processing conferences of biologics that I've attended, and they just need to see it done well and done according to the guidelines that they have about what they expect for a protein therapeutic. But they like the idea of less in the way of stop and start. They like the idea of moving along, and because when there are hold times in a process, these sensitive biologics can degrade. And by continuously producing it, if you think of what the molecule sees going through the process, it's in the biological tank for a while, being produced by cells, and then if it's continuously pulled from that and captured in the first column, passed to the second, goes through some filters, etc., it's going through a very consistent path. And there's a lot of support for this at the FDA. I can't speak to the EU and other authorities, but at least in my interactions with FDA, they like the idea of continuous process.
To me, the process I'd rather the process to be in stasis is the dream, right? I mean, this thing, no matter what, that is where you want to be. The stop and start, the batch, that's the danger. So I mean, that's where the problems are.
It's gonna be batch sometimes, batch, which is that perfusion. I don't know, but perfusion will come as well. But it's going to be a hybrid at times as well. Not everything is going to line up and be continuous start to finish, all in one go for every single molecule. So companies and process engineers and chemies and rest of it, biochem, need to look at it more about it's a jigsaw puzzle, and we can put it together and then work out what is best for that particular molecule. And as cell therapy and gene therapy start to kick in as well, that's going to go through a similar sort of like evolution. And again, as those personalized medicines and smaller volumes, we've got to start thinking differently about how we put those processes together.
The advantage I think we got here, and Hallil, I wanted to come back to this. I mean, we could look over their shoulders in Europe. I mean, if it works, I mean, guys, sum it up. I mean, what can we copy to make it move along here?
Well, one of the things is the regulatory pathway has been well accepted. There is a presumption. There is no longer presumption in Europe as we had in the U.S. right now that these products are dangerous. Some people think switching is dangerous. Well, we haven't seen that. We've published on that extensively. The review came out a month and a half ago, 90 studies. No one has seen these dangers. But so what they have is two things. Health authorities are now willing to accept them, and they have initiated education programs. Ireland, Netherlands, France have now initiated. So it's not just having the products get approved, but it's also integrated with an education program to educate the doctors and the patients about the value. And what we've actually found, and there's been published just a couple of weeks ago, In fact, we see that in the U.S. in the Carolina Oncology Group, where the money saved on biosimilars has been poured back into patient services and patient access. So this is something that we can learn from Europe, that the pathway is viable, that we have to have high standards. There are actually the initials, if you take a look at the initial rounds of products submitted in Europe, there were quite a few rejections due to it did not meet comparability. In fact, I'm not sure some of the first biosimilars that were licensed in Europe would meet today's standards. So the standards of science have been accepted, and I'm glad to see that the World Health Organization is accepting that as well. So that's a good thing. And the integration of the education is also something I think that we should look at as well. Now the FDA is beginning to copy that. There is a consensus document that was done in Europe of patients, payers, health authorities, industry. We don't have anything like that in the US. I think that would be beneficial. Instead, we have a hodgepodge. Now, granted, everyone is moving along, but to have that integrated whole, I think, would be beneficial. So I think integration of educational activities would be something very helpful.
Thank you, Hallil. I thank our panelists. They're not running away, so tackle them as they leave. Thank you, gentlemen.
You are
That's
right.
it.
Thank you very
Okay.
much.
Pharmaceutical Processing, in conjunction with Interfex 2018, presents Interfex Live.